Now let's talk about the structure of F class pump in the inner mitochondrial membrane. As you can see, this is the intermembrane space where the hydrogen or proton concentration is high, and this is the matrix where it's low. And there is a charge separations. Inside is positively charged and outside is negatively charged. That means the matrix portions is the negatively charged. So protons actually move from intermembrane space to the matrix, and that's how it generates these portions of F1. It actually generates the ATP. Now at first I want to mention that this FO, O for oligomycin sensitive, which is not the 0, it is FO, O for oligomycin sensitive. This subcomplex contains 3 proteins. One is A, B, C. See here, this is the A portion, this red is the A portion, A protein. This is the B protein, and this C is actually subunit, and it makes a ring-like structure called C ring. So in the FO, there is ABC. In case of bacteria, bacterial F class pump, there is 1 A protein, 2 B protein, and 10 C subunit makes the C ring. So there is a 10 C subunit in case of bacteria. The each mitochondrial F class pump, the FO portion is very similar with the bacteria A1, B2, C10. I am actually mentioning the structure of mammalian mitochondria. That is A1, there is 1 A, there is 1 B, and C8. There is 8 C subunits that makes the C ring, and this C ring actually works like a turbine. It actually rotates, and by these rotations, actually ATP is generated. How it occurs, I will mention. And F1 portion of this protein contains alpha beta subunit that present in hexamer. That means there is 3 alpha and 3 beta alternatively placed with each other, and it makes the hexamer. There is a gamma portion of this protein. This rod-like structure is the gamma. There is a delta structure that attached with 1 alpha and attached with a B, and there is an epsilon that present here. So in this structure, there is 2 actually conditions or 2 situations. One is called rigid and it is rotor. That means this structure, in this structure, some portion of the protein actually rotates and some portion remains actually rigid. The rigid portion is this A structure, this B structure, the alpha beta 3 hexamer, and this delta. So these portions is actually rigid. They do not move, they do not rotate. But the rotor is the C ring, the turbine, the gamma, and the epsilon. Now I want to mention here that this B subunit of the FO makes a tight contact with the delta and A. Okay, they are very tightly joined. The gamma and epsilon with the C ring, they tightly join. And this hexamer is actually positioned at the top of this gamma rod. Now the gamma rod is actually called the rotor stalk or central stalk. We also call it rotating shaft, like the gear. And this B subunit is called stator stalk. That means stator means this is static, not rotating, or peripheral stalk. You can remember any of this. Now let me explain how the movement of protons, that means through the proton motive force, the protons actually move from the intermembrane space to the matrix, and how the movement of protons actually helps in the rotation of the C ring, and that's why it is called turbine. It actually rotates, and that rotation is actually causing the rotation of this gamma rod-like structure, and then this gamma rod-like structure actually changes the structure of conformation of beta subunits. Remember this. So there is a sequence of things actually occurring. Proton movement actually is the first thing, and proton movements occurs due to the PMF, proton motive force. So for the first point, we have to remember that there is a proton motive force. That proton motive force helps in the movement of protons. So proton moves through the C ring, and that's how C ring actually rotates. These rotations actually cause the rotation of the gamma rod. This is a rod-like structure. Then movement of gamma actually cause the conformational change. Conformational change in beta. And that structural change of beta causing the ATP and PI to make ATP. So basically, in the alpha portions of this alpha beta hexamer, ATP can bind or ADPPI can bind. So ADPPI can bind here, but alpha do not directly involve in the ATP synthesis. Binding of ADPPI or ATP in the alpha just maintains the rate of ATP generations. The main function or main ATP synthesis is performed by these 3 beta subunits. Now, first thing we have to understand, I have actually mentioned what is PMF, and now we have to understand the second fact. So this is the first point we have to understand. How ATP is generated? The ATP is generated by the conformational change of beta protein, but the start point is here, the PMF, proton motive force. Second, proton actually moves through this seeding, and seeding actually rotates. So this rotation rotates is the third step. Then the gamma rod rotates. This is the fourth step. These gamma rods mechanically, remember this, just like we push someone, mechanically it can actually change the conformation of beta. So this is the fifth stage, and then ATP is generated. This is the sixth step. So I have just mentioned the proton motive force and the structure. Now we have to understand the how proton moves through the seeding, and then seeding rotates and the gamma rotates. Now by this diagram I can easily explain how protons move through the seeding and ultimately release into the matrix of the mitochondria. Here two structure is important. One is called proton half channel 1 and this proton half channel 2 that present in the interface of protein A and C subunit. As I have mentioned, all the C subunits are actually contain the negative charge amino acid aspartate. In case of mitochondria it is called aspartate 61. And this negative amino acid is actually responsible to bind with the protons. Now, if you zoom this seeding structure, you will find that protons actually move through this half channel 1, and the C subunit that is close to the half channel 1 actually empty. Now, as because there is a negative charge amino acid aspartate 61 is present, the negative charge is neutralized by another amino acids present in this A protein that is called arginine 210. These amino acids actually neutralize the negative charge present in the C subunit that close to the half channel 1, so that when protons enter, these protons actually displace the arginine 210 into the C subunit that is close to the half channel 2. So when arginine 210 move from this half channel 1 to the half channel 2, the protons that is present in the half channel 2 is actually displaced or expelled into the matrix. And when the protons is actually expelled into the matrix, there is a rotation occur anti-clockwise, and then this empty site, actually the C subunit come close to the half channel 1, and the cycle continues. You can also understand the cycle of movement of protons by this diagram. Here the arginine 210 is not shown, but aspartic acid 61 is actually shown here. One important point I want to mention that if you modify this aspartate 61 with a poison called dicyclohexyl carbodimide, so DCCD, or if you mutate this aspartate with alanine, as because alanine amino acid don't have any charge, you will find that the proton pump actually stops. So this aspartate 61 is so important. Okay, so here as you can see, protons enter through the half channel 1 and it go through all the C subunits, and ultimately it will release from the channel 2 into the matrix. To understand this, I have zoomed this seeding structure only and this gamma. The first thing we have to understand that in the intermembrane space, as because proton is high and here is low, so proton will move from this seeding to the matrix side. Now in between this A protein and the seeding, these are all C subunit. Remember, there is 8 C subunit present in the mitochondria. In between this A and C interface, there is 2 half channels present. One is called half channel 1 and another is called half channel 2. Now why is it called half channel? Because through this channel or half channel, proton cannot directly move from this to here to the intermembrane space to the matrix. It cannot go like this. Okay, that's why it is called the half channel. Proton can enter through the half channel but cannot go out through this second channel. It can enter and it can go into the first C ring or C subunit. This negative sign, what is this negative sign? In every C subunit, remember this, in every C subunit, there is a glutamate or aspartate amino acids present that carry negative charge. In all the C subunit, there is, whether it could be a glutamate or aspartate. In case of mitochondria, it has been found aspartate in the 61 positions that gives the negative charge. Why it is important? Because in every C subunit, protons will be accepted and proton carries a positive charge. And this aspartic acid or glutamic acid present in the bacterial F class pump, suppose this aspartic acid, these amino acids can actually arrange for the protons to come inside of this subunit. So every C subunit contains this aspartic acid or aspartate here. But one thing we have to understand, that except the first C subunit, in this C subunit, there is another amino acid is present that carries positive charge, that is called arginine. This amino acid carries a positive charge. This arginine can move, this is actually a flexible amino acid, it moves from first C subunit to the last C subunit, remember, in the C8. I am mentioning the number, but in the real structure there is no number, there is no C1, C8. We are actually denoting this number to understand it easily. So suppose this is the first C subunit, basically it actually rotates, so there is no 1, 2, but suppose the cycle starts here, starts from these situations. Then in this C1 subunit, there is no proton because the proton is just entering inside this empty portion. So we call this C subunit empty proton binding site. So the first C1, I will call it empty proton site. So in this C1, there is no proton present. Now, I have mentioned that every C contains aspartic acid, negative charge. Now, who will neutralize this negative charge in this C subunit? There is a positive amino acid that is called arginine. These amino acids are actually fixed or actually fit inside this C1 so that the negative charge of this aspartic acid can be neutralized. But what happens when a proton enters through this half channel 1 to this side, then this arginine actually repelled. It actually repelled because one proton, say a positive charge is entering, and that will repel this positive amino acids. Now when the first, suppose this is the cycle, this cycle starts from here, when the first proton entering through this half channel 1 to the first C1 subunit where there is no proton, then this arginine amino acid actually swing back into the C8 of the subunits to remove the proton that is present already. So in the C8, there is already a proton present, and when this arginine moves from this C1 to the C8, suppose this arginine is moving from here, this amino acids, then it actually repel the proton to move from this push C8 to come out from the half channel 2. And by this way, when protons move, what happens? Actually rotation occurs. So when the protons are actually empty, because arginine actually empty these protons, then arginine present here. So there is only this aspartic acid negative charge present and the arginine positive charge is present, and this is actually empty conditions. See, this is empty condition. So when this C ring will rotate, this 8 position C8 will become the C1. So C8 will become the C1, and then another proton will enter into the empty site and actually move another time the arginine to the C8, and then another proton will move. So this C ring actually acts like a turbine, and the proton motive force is the first factor that actually helps in the movement of H plus ions through this C ring, and with the help of A protein that causes the rotation of the C ring. And as because this gamma, that is the rotor stalk, is tightly attached with this C ring, so movement of C ring is actually rotation of C ring causes the rotation of gamma. Now the gamma rotation can cause the conformation change mechanically, just like mechanically it actually changes the structure of the beta. Now this is actually proposed, this hypothesis was proposed, the conformational change of beta, by Paul Boyer, and this hypothesis is called binding change hypothesis. Now or mechanism, binding change mechanism. Here the binding means binding of ADP and PI, and change means the conformation change of beta. So binding of ADP and PI and change of beta conformation is actually main theme of this mechanism. This hypothesis was proposed by Paul Boyer in 1979, but in 1994 John Walker at the Medical Council Research, he actually discovered through the X-ray crystallography the exact nature of beta subunit conformational changes. He observed that these beta subunits goes through three conformational changes. One is called O state, another is called L state, and the last is called T state. And then T states actually go back to O state. Now O means open state, O means open state, L means loose state, and T means tight state. So the every beta subunit goes through this O state to L state to T state. What happens here? In the O state, this beta subunit, it could be any beta subunit, all these three beta subunits go through this. So in O state, beta subunit can bind with ADP and PI very weakly it binds, and it release ATP. In the L state or loose state, it bind ADP and PI strongly, but cannot bind with ATP. And in the T state, it bind ADP and PI so tightly that it may or actually generate ATP. So in this T state, ATP is actually produced, but this T state, ATP cannot be released. Remember this, ATP cannot be released. So it is in lock state. So what I am talking about, this 3 beta can go through these 3 different conformations, and these conformations defines the affinity towards the ADP and PI. Mechanical, this gamma actually rod actually rotates, and it causes the mechanical force to change the conformation of every beta subunit into O state to L state and T state. Suppose this beta, suppose this is the beta 1, this is the beta 2 and beta 3. I'm just giving the name. This is actually going through O state to L state and T state. Suppose this is the cycle. It is also going through O state to L state to T state and back to the O state. It is also going to do O state to L state and then to the T state. So what happens if this is the first beta, let's suppose, then if this beta is in O state, the second will be in the L state, the third will be in the T state. When the gamma rod rotates, suppose the rotation is present here, then when this gamma actually rotates in 120 degree, because the complete rotation is 360 degree, for 3 beta to touch with this 3 beta, every 120 degree rotation can cause the conformational change. So when 120 degrees rotation of this gamma occurs, then this beta, that is beta 2, will change from L state to T state, and this T state will move into O state. And when the beta will go into the O state, in the T state, the ATP is generated, will be released. So ATP will be released in this state. Now what happens when this beta is actually L state? It then go to the T state and it actually generates the ATP, but it actually holds the ATP. And this T state will actually, here ATP is produced, but when it go to the O state, it actually release the ATP. So what happens in every 120 degree rotation of gamma, every 120 degree rotation of gamma, 1 ATP will be generated. And with the 360 degree rotations of the gamma, 3 ATP will be generated. And do you know how much revolution occur in 1 second by this gamma? The gamma actually revolves 134 times per second. And in 1 revolutions, that is a 360 degree, it generates 3 ATP. So for 1 second, it will generate 134 into 3. That means approximate 400 ATP will be generated in 1 second through these rotator machines. This is very unique machines. So through this conformation change, it actually causes the binding of ADP with the PI, that means the terminal ATP. So ADP binds with the PI, this is the terminal phosphate, and makes the ATP.